Hello, everyone, and welcome to today's STERIS Tech Talk on successfully validating a sterile barrier system. STERIS Tech Talks are a series of webinars covering subjects relating to gas and radiation sterilization processing and the laboratory testing and validation services which support these processes. Our presenter today is Noel Gibbons.
Hi. Thanks very much, Ashley, for the introduction. My name is Noel Gibbons. I'm a technical advisor on packaging for STERIS. I've been with the company for just over 23 years and have worked in various different functions. In relation to STERIS, STERIS AST, or Applied Sterilization Technologies, support medical devices and pharmaceutical manufacturers throughout a global network of contract sterilization and laboratory testing facilities, of which we have a total of 60 locations worldwide. We ensure that the patient safety through the delivery of sterile single-use healthcare products. In doing that, our core offerings are contact sterilization, testing services, equipment and technologies, dosiometry, and technical professionals. In relation to product and package testing, we have two prime locations. The main one in Galway is where I'm based, and the second one is in Brooklyn Park in the US. The testing services that we offer basically cover product and package testing. So these include accelerated and real-time aging, medical, mechanic, or electrical equipment, accelerated lifestyle testing or life cycle testing, distribution and transportation simulation, extractables and leachables, lure connectors, cold chain testing, shelf life, an extension of shelf life, sterile barrier integrity system, product validation and design verification of products and packaging. So the question that always has to be asked is, why do you do anything? And in this case, why do we do package and integrity testing or string testing? So the real answer to that is a number of ways or reasons. And it's basically essential to assess the product safety of the actual product and the packaging itself to ensure that it retains its integrity after the transportation or throughout its shelf life. Because if it's not able to do that, then you're going to run into trouble at the end of the product's life cycle, where you look at the very bottom of the screen, it's in relation to patient protection, where you will not have provided that patient protection to prevent potential harm to the patient. We also have, say, the likes of the regulatory requirements and standards requirements, such as ISO 11607 and in Europe, the MDR. We also have the quality assurance to maintain the quality of the product by identifying any weaknesses in the packaging. And as I said, to protect patients from potential harm caused by integrity breaches and therefore possibly infection due to that process. What are we testing? Basically, a sterile barrier is what we're providing in the packaging once it's been through the sterilization process. And if you look at, say, what the definition of a sterile barrier is, it's the minimum package that provides the minimum risk of ingress of microorganisms. Basically, it prevents contamination. It allows aseptic presentation, and this must be maintained throughout the sterile life of the packaging to its point of use. And by point of use, what we mean is when it's opened, either at the patient's bedside or wherever it is actually going to be opened and used. The one difficult thing that a lot of companies have here is where and how they identify what is the sterile barrier. If we take, for example, the product we have in front of us, it's a pre-filled sterile syringe, okay? So it has its own sterile barrier built into it. But you'll find in many occasions that these syringes are also packed into other packaging, such as, say, the tray with the likes of a Tyvek or a paper lid on top of it, or foil lid, depending on the product. But the problem is here, which is the sterile barrier. Is it the tray, or is it the actual blister? Or sorry, the syringe, I should say. And that is dependent or down to the customer or the owner of the product to determine what that is. When we're looking at validating the sterile barrier system, there is a fairly significant process that it should go through. And that should go all the way from DQ or design qualification through installation qualification, operation qualification, and process qualification. But we primarily look from the OQ stage onwards, because that's where we introduce the actual seal strength testing. And that's when you are establishing the seal strength of your packaging and looking for the nominal seal strength. Then you move on to PQ, where you introduce the package integrity test, whether it could be bubble leak, could be dye pin, it could be bubble leak under vacuum, could be microbial type testing. That's again dependent on the packaging and the product itself. And that also should also include seal strength on that. And as you can see from the flow chart here, that goes all the way right across each of the processes, through the production, through sterilization. You then have the option to do either baseline testing or go directly into transportation testing. But the most important thing you have to do there is take the results, particularly from the seal strength testing, and be able to compare those back to your OQ settings and sealing parameters, because then you can show that the seal strength has not deviated significantly over the entire shelf life of the product. When we're looking and talking about design qualification or the phases of validation, what we're looking for is objective evidence that the requirements or the user specification meets the actual supplier specifications, that the functional spec matches the actual supplier specifications for the materials, that you can carry out a factory acceptance test and how that's done, and verifying the packaging material that it is actually correct. The same thing applies for installation qualification, again looking for objective evidence to ensure that the correct installation of any hardware or software products that are required to create a sterile barrier. And again, a site acceptance when you get your product and your packaging on site to make sure that they can actually be used and in the format and that you have defined. In relation to the OQ, again, you're looking to establish the sealing parameters here on temperature, pressure, and time to create the actual sterile barrier. And from that, you should be able to do the likes of packaging tests and performance tests of the actual product itself. And again, for PQ, or product process qualification, again, objective evidence is what is required. There's also post-validation, where you're looking at maintenance of the validation and how you do that. So like the revalidation of equipment, a reassessment of materials and parts and the packaging materials, and also the manufacturing process and how you do that. In relation to the regulatory bodies, what are they looking for? They're actually looking for you to prove that the overall integrity and safety of the packaging is in place. And that's done by subjecting the packaged product to rigorous validation processes. And these would include the likes of your packaging process, full validation, your stability testing, including real-time and accelerated aging, performance testing through distribution simulation, and package strength and package integrity testing, basically showing that you have maintained the integrity of the product, of the pouch or tray or whatever the case may be, and also the strength of the barrier itself. There are a couple of challenges there when you're doing that, and one of them is basically designing safe packaging that will produce or protect the medical device during the shipping and transportation of the product to the point of use. And the second challenge is to actually check or test to a particular standard and defining what that particular standard may be. If you look at, say, ISO 11607 Part 1 in Annex B, there are 123 different standards listed, covering 34 different types of tests. And you have to make your decision on which one of those based on your risk assessment and a justification for each of those test standards. Say, for example, if you're to look at distribution testing, you'd use the likes of ASTM D4169 or ISTA 3A or 3E. If you're looking at the integrity of your pouch, you'd start with a visual inspection to ASTM F1886, and then do the likes of a dye penetration to F1929 or 3039, and also then F2096 is your bubble leak testing, followed by either a burst test or a peel strength or seal strength test to the likes of ASTM F88. There's lots of different types of tests out there, but if you look at the USP, what that actually does, it defines testing as both probabilistic or deterministic. Probabilistic methods provide results based on statistical probability rather than deterministic outcomes. And by that what it means is basically a lot of them are pass-fail. There's very little measurement in them. The typical test that we would do on packaging there would be the likes of dye penetration testing, bubble leak testing, or seal strength testing or tensile testing. Interestingly enough, tensile testing actually falls under both camps for a very specific reason. And that being that you have variability in the materials, you have variability in the processes, but you can also have precise and repeatable results. So that would also fall under the deterministic view. So the deterministic methods provide precise and repeatable results under controlled conditions. These include the likes of vacuum decay test or pressure testing. You're doing chemical characterizations, particulate testing, extractables, leachables, and all the tests that are associated with those. But you will notice that the difference in both of the test types is that the probabilistic are more aimed at the outer packaging, whereas the deterministic are more aimed towards the actual product and the internals of the actual packaging itself. So again, when you're actually looking at the actual test methods, which one do you actually choose and how do you choose it? Basically, you look at a very early stage literature review to determine what standards are there and what's applicable to your product. And as I said, 11607 Part 1 Annex B actually has a full list of standards that will be applicable to the majority of products being placed on the market. You can have device test portfolios which contain both standard and non-standard tests. Non-standard tests are more difficult to get through the process because you have to validate and justify why you're doing them. And some devices will require more extensive testing and method validation than others, along with additional method creation. Packaging design and labeling, again, it's very important that you understand the labeling concept that come along with a sterile device. You need to look at everything from the instructions for use guide, or the IFU or DFU, whichever you call it, or EFU. It also has to contain, say, the likes of the batch numbers or lot numbers, expiration dates, handling or temperature requirements, and very specifically, when I'm talking about that, I'm talking about specific storage conditions. Again, you need to be careful when you are placing that, because it may have implications for your accelerated aging and the temperatures involved used there. The intended sterilization process, no matter which sterilization process, it has to be indicated on the actual pouch or tray or package, as the case may be. And any permanent or removable labeling, so like the patient labels, which would normally be removed and placed on their charts in a hospital. There's also very specific details on any product that may contain materials that require a cautionary statement for likes of latex or any other materials that can cause a severe or even mild allergic reaction. And any languages, how many languages? But the problem with the IFUs is they've gone from 1 or 2 pages to anything up to 4, 5, 6, 7, 800 pages. So the IFU is getting larger and larger. In relation to the sterile barrier symbols, when there is a requirement within both the MDR and ISO 11, which basically states that where the SBS is to be opened at the point of use and consists of more than one packaging layer, the sterile barrier system shall have an indication to be recognized as such. Basically what that's saying is that the person that's opening it has to be able to identify which is the sterile barrier and what level, if it is, if it's multiple packaging. And a lot of companies still don't actually use this, which is very important actually for the user to be able to identify. And it's very simple to add on to your labeling system. There are lots and lots of other different types of labels as well, which actually shows you whether it's the sterilization, the temperature range, the likes of where it is made, all have to be there. And the likes of serialization, all again should be there in relation to the packaging itself. As I said, in the additional samples that have been, or symbols that have been added to ISO 15223-1, and there was 21 of them in total. But again, it shows you the oval symbols in relation to the packaging levels, which are defined by the Sterile Barrier Association and included within this standard. Again, in relation to failures on labels, what are the failures on labels and what can they cause? Basically, if you have a torn or illegible label, if it's not human readable, it's going to cause a major issue because you don't know if you've got the right expiration date, if you have the right product. If in relation to the labeling itself and how it's created, if the materials are not compatible with the ink, if they're not compatible with the actual whole material process and how it's printed, then it's going to basically fade very quickly and it'll cause the ink and to basically rub off as soon as it's touched. In relation to package integrity testing, there's lots of different tests out there that'll test to lots of different levels. And what you have to decide on what's practical and what's effective for your packaging. Take, for example, if you look at, say, the likes of bubble leak testing, that has been validated down as far as 250 microns. Whereas if you take the likes of dye penetration testing, that has been validated down as far as 50 microns, depending on which standard that you're actually looking at. You can go a lot lower than that and go down as far as maybe 1 micron, depending on the likes of gas sniffer tests and things like that. But again, that's dependent on what's available and what's applicable to your packaging and the materials that you're actually using. The typical test standards that are normally used for validation in medical devices will be the likes of inspection. So that's be a visual inspection, bubble leak testing, dye penetration testing, or peel or seal string testing. If we look at, say, the likes of the visual inspection, this is done to a standard called the ASTM F1886. And currently this standard only looks at the seals of the actual pouch or tray, as the case may be. But you can detect a defect down to approximately 75 microns in that with a 60 to 100% probability. Again, it's a qualitative measure. And again, it's, as I said, it's on the unopened seals to determine the presence of defects there. It's likely to be directed to variables within your process and will show you issues within your process, showing whether you have a problem with your sealing machine. If it's a material issue, it can point you in that direction and very early, and it's a very good test to have on the end of a line. Take, for example, the graphic that I have here on screen. This is actually a representation of a defect that's quite common in pouches and trays, where you're getting a tortuous path shown or coming through the actual product, which actually causes a breach in the sterile barrier. As you can see, there's a full breach of the seal here. And this is actually a trace of a drawing of an actual pouch and defect that we found in one of them. What you're looking for with the standard is basically you're looking for non-uniform seals, foreign particles or dirt or debris in the seal area, transparent areas where the material is actually being burnt, wrinkles or visible bubbles throughout the actual seal, or channels in the seal. The current standard is currently being under review and is being updated and will eventually include tests for the surface of the pouch as well. But the problem is, what happens if things go wrong and you do start to find defects? So for the likes of an open pouch, something that was never sealed, and this gets to the end of your line, what do you do? Basically, if you have seal breaches or foreign particles, cracks, tears, or the reject level is much higher than you would expect in normal, What do you do? How do you get around that? How do you actually investigate? Do you go back and do you look at and investigate the root cause, risk assess it, the test results, retain or retrain the operators? Again, it's down to you as the manufacturer of this sterile barrier to determine what to do and how to fix that problem. But again, it's something that you need to be aware of and keep in your mind that this can happen. Typical visual inspection failures, so the likes of fibers in the seal, or the product actually encroaching into the seal, whereas in this instance in picture number 2, it's left less than 1 millimeter, approximately 1 millimeter of seal left, and one good bang and that's gone. So your entire seal is breached. Seal creep, again, is another major issue where the seal is not strong enough to actually hold the product inside and the seal starts to open. Where you have oversealed pouches or seals, basically the material has started to melt and become transparent, indicating that it's now going to start to become brittle. You have channels in the seals, pinholes in seals, tears, cuts, rips. Again, you can have all of that in any part of the actual pouch itself. One of the big problems is where you have hidden dangers that you don't see and are very, very difficult to detect. So if like on the first picture there on the left-hand side, there's a sharp burr, approximately 1 millimeter. And that actually caused a slit or a cut in the actual sterile barrier, the pouch that it was placed into, which is the third picture. And the only place that was detectable was during a bubble leak test. Because it was so small, the defect couldn't actually be found during a visual inspection. So again, looking at the whole process and how that tray was made and how that was cut off is very, very important to have that under control. So like a failure in foil pouches where you're getting a lot off, so like a bending in a heavy foil, it'll act, say, like the likes of a drawing pin. If you spin it back and forth and back and forth and back and forth, it'll eventually break. And what it can cause is actually holes through the actual pouch as well. Peel strength or seal strength. Again, what you're looking to do is you're looking to have a validated process for your seal strength. And this normally goes through what I call a 3-cubed process or 2-cubed process for your temperature, your pressure, and your time, where you're actually testing the 3 main parameters at all the different levels. So it's your max temperature, your min temperature, your max pressure, your min pressure, and your max time and your min time. So basically what you're doing is you're getting a set of samples that can be tested, and what you're doing is you're looking for the actual nominal setting for that particular pouch and those particular materials. And that's what you're looking for, is that you're in the middle of that range. If you're towards the spotty seal, this means that it's most likely going to be opened or very easily opened. If you're on the glazed side, it's going to be basically what's going to happen is that it's going to be burnt and that will possibly crack open during the use of the actual pouch itself. Again, what you're looking for is looking to get on the graph on the bottom here is on the flat line area. You're actually trying to get someplace in the middle there. And that is basically an output from a seal strength evaluation that we did some years ago. In relation to sampling, none of the standards will tell you really that you should take samples from this side or that side or where you should take them. The only one that does that would be the likes of EN 868, which recommends that you take a minimum of 1 per seal and that you treat the chevron as a single seal. So you're not taking 2 samples for that. And that anything that's over 500 millimeters, you should look at taking extra samples. So what we've put together and agreed with most of our customers that we actually work with is that for particular size of a pouch, if it's greater than 350 millimeters, we will take 2 samples. If it's greater than 500 millimeters, we'll take 3 samples from that particular side. And that's how we work out the sample numbers. Again, it's totally up to you as the customer to define how many you want. But the minimum that I would recommend would be an absolute minimum of 1 And that minimum could be actually on your own closing seal, but that's totally up to you and your risk assessment. In relation to peel testing, you can see the video that's running here at the moment. All it's doing is basically you cut a sample out of the pouch and you put it into the jaws of a tensile tester and press start. And that will give you the details on the actual seal itself, which we'll look at shortly. Again, what you're looking for is you're looking for consistency and repeatability. When you're doing the testing, it depends on the materials that you're using, the company you're working for, the country you're in, the size of the actual sample that you're using. Typically, European countries use 15 millimeters, the US typically uses 1 inch, and that's going back to historic times. Again, what this is doing is providing you variable data on the force and the characteristics of the actual product itself. F88 itself was updated recently to include the likes of rigid packaging or trays, which it didn't typically cover before. So you now have included 2 additional techniques in technique C, which covered the trays, and you can actually test them either by cutting out the actual sample out of the tray or test the sample within the tray itself. So what you're doing there is basically you're using the tray to create the backing or the support to the actual seal itself while you're doing it, and basically replicating the original technique C. You can see this in better detail here, where you're actually cutting through the actual sample for technique C and actually removing the sample out of it. Whereas in the second one, you're leaving the tray intact and using the tray to actually give you that support. Again, what you need to look at what happens if something goes wrong. If you get an open pouch, if it's very badly sealed or partially sealed pouch, what's happening to it? If the peel level is below what you expected, if it's above your accepted, then you have more problems. If the CPK value is outside the accepted levels, how do you actually address all of those? There are other occasions where you need to look at the actual test method that has been actually used. Take, for example, if you use, say, the likes of technique A, and you're getting pretty much test level of in around 7, 8, 9 newtons. If you test the exact same samples using 180 degree supported, you'll get 18, 19, 20 newtons from the same samples, from the same pouch. But if you use technique B, you get a significant lower level. So you need to be careful on which methods you're actually doing. And also, if you're looking at, say, the likes of different lengths or sizes of pouches, the graph on the left-hand side there is actually from a series of pouches that were approximately 2.5 meters long. The bottom graphs there was actually part at near the closing seal. The middle ones were from the end of the pouch, the chevron end, and the top ones were the center of the pouch, again showing the different seal levels across the different pouches. But what you're looking for there when you're doing the test is the transfer between the actual different materials to see if you've got a good sample or not, if you have good transfer or not, which will basically tell you how good or bad your actual seal is. The percentages that are given here on screen are guesstimates, give you an idea on how good or bad the actual seal is. And it's again up to you during your validation, put that in practice. Packaging integrity penetration testing. Again, there is a couple different types of test. They can be quite subjective because they're extremely fast in that they will detect a defect within 1 second. And they will start to wick and leach out of the actual pouch, or the dye will start to wick and leach out of the pouch within a couple of minutes, and it may actually hide the defect. But it's a very good test. It's very fast in that you can actually test an entire pouch within 20 seconds, depending, of course, on the size of the pouch. The intent of the standard is to detect channels across the entire seal. Sometimes you will get partial channels across the seal. These are not defects because you still have integrity, or the integrity of the actual pouch or tray retained. Typical example of a defect in a tray, what's shown here is 125 microns versus 250 micron. And you can see that the dye will actually pour directly out straight through the defect, and it can disappear within a couple of minutes. The picture on the right-hand side is an actual defect that was found in a customer's pouch, and you can see how very clear that is. The problem, as I said, is the wicking and the leaching of the actual dye through the materials. The tray that's actually shown in the pictures, in both pictures here, was approximately A4 size. So you put 5 millimeters dye along seal and you run it around each of the seals, and you can see where it is after 1 minute and after 3 minutes. So any defect that was in there will actually disappear in a very, very short period of time. Again, looking back at the defect we've seen during the visual inspection in the seal, we can see that will run straight. But one problem that is there is that you can also get false positives, which you need to be very careful of. In the picture on the right-hand side, this is actually a false positive where the pouch had actually been folded and creased and created a path that the dye could actually flow through, but it was not an actual defect itself. Bubble leak testing, again, a very, very good test, in that it's a whole package integrity test. So it not only tests your seals and your surfaces, but absolutely everything that you put the pouch through. So it will assess everything in any of the tests that you've put it through, or any of the mechanical type tests that you've put it through previously. It can be done on either porous or non-porous packaging. And that includes the likes of Tyvek and paper. Paper can be extremely difficult to test depending on the grade of paper, and it is very, very fast test again. It's used to test by either pouches or blister packs and trays. But as I said, its disadvantage is that its sensitivity is only down as far as 250 micron, which is pretty much the equivalent, the thickness of a human hair. And again, it depends on the pressure that you can get in there. So for the likes of Tyvek or paper, they have their own natural breathing points, which are in around 2 to 2.5 kPa, possibly 3 kPa, whereas a defect around a 250 micron will be detected in between 1.5 kPa and 2 kPa. So it's quite easy to detect. Again, it can have issues in that it can show up false positives. If you look at the video that's playing at the moment on screen, you'll see that this is actually what you're looking for. It's a continuous stream of bubbles out through a defect. But you can also get those continuous streams of bubbles from other areas in the pouch. So you have to be able to distinguish what they are. And basically the very simplest way to do that, if you're getting a continuous stream of bubbles from the likes of a pouch, a Tyvek or a paper or pouch, all you do is rub your finger across the top of it. If they stop momentarily, it's breathing. If it doesn't stop, it's more than likely a defect. The problem happens though when you start to get defects on the side or the seal or through the seal. Which are they actual defects, or are they basically from where the pouch has actually been folded and creased, which we'll have a look at shortly. Again, if we look at the what ifs, what happens if you have a pouch opening during testing, which does happen quite often. That means tell us that the seal is very, very weak and that it wouldn't even pass the likes of a burst test, that its seal strength would be very low. If you're getting punctures, if you're getting trays cracked, which is very common, depending on whether they're from the final molding or from an engineering mold. Again, you see lots of different types of defects coming through from the various manufacturing processes. As I mentioned, that the escaping bubbles basically from the seal, that can be caused for the likes of where the material has been folded over. So you're getting sheet separation or sheet bulk density reduction. And what that's doing is basically it's splitting the material at a certain point throughout the actual seal, across the seal. So if that splits at in the middle of, say, the Tyvek or paper, that'll be fine for today because you still have a sterile barrier. It may be fine, it may be accepted by your company, it may not. That is totally up to you. But if it does split at the actual clear film side, then you definitely will have a problem because you have a 99% chance of having a breach in your sterile barrier. Seal strength or burst testing. Again, it's a very good test, but it's only a snapshot of a test because once it's burst, it's gone. You can't repeat anything with it. Unlike, say, the likes of peel testing, where you can repeat the seal strength all the right way around the whole pouch, no matter, as long as you have real estate to actually take a sample from. You can take another sample and repeat that test. However, with burst testing, there are a couple of different methods with that can be used. The ASTM F1140 is a non-restrained method of the test, and that has a number of sub-tests as well. So you have the unrestrained burst or dynamic burst, or you have a creep test or a creep to fail test. The big problem with this type of test is that you have the effect of what's called pillowing. So what that does is basically when you blow up a pouch, it creates strain on particular areas of the pouch. And those areas may not be the actual weakest point of the pouch, but they will more than likely burst in those areas. So you need to be very careful on how you're doing that. If we look at, say, the other version of the test, which comes under ASTM F2054, it's a restrained test. And what that does is significantly reduces the pillowing effect of the pouch. And reduces those, or virtually eliminates those strain areas or stress areas on the actual seal, and you have a much better chance of detecting the defect, or sorry, a weak seal in that. Again, what you're looking for in either of the standards, it's consistency and consistent and documented results, and whether it's your own in-house seal or it's a manufacturer's seal that's actually bursting. And the one thing that you continuously need to do is monitor these results and monitor them over time. So every batch that comes in, you need to be able to do a burst test on it and make sure that it's repeatable and that it's within your limits that you have set for that in your validations. Because if it's not, you're eventually going to get to a stage where you're no longer going to be able to use the actual pouches themselves. Bubble leak test under vacuum, again, it's another very good test in that, again, it's a whole package integrity test, and it can test the likes of the foil and foil type packaging or non-breathable packaging, and it's very easy to do, again, because all you're doing is basically submerging the product under water, and you're applying a vacuum to the test and looking for a continuous stream of bubbles from that actual pouch as well. Again, it's for the non-porous pouches. It doesn't work with porous type pouches. There are a couple of different types there that you can use, so the likes of the ASTM D3078 or alternatively ASTM D4991. D4991 is not specifically for medical pouches or pouches. It's for rigid type packaging, but again, it can be adapted to use in the likes of pouches or trays or anything that's rigid. But again, it's down to you to determine what that is and validate that for your particular packaging. You can basically detect different size defects depending on the different types of pressure or levels of pressure that you can actually apply to the actual package itself. Typically, you will find a 100 micron diameter defect at around 22 millibar. 60 micron at 50 millibar and 25 millimeters, someplace between 400 and 700 millibar, depending on the material that you're actually testing and the pouch and the size, the temperature of the water. If you're using, say, the likes of Tripenex or anything like that in the actual water to actually reduce the viscosity of the water, again, that's again totally up to you, but you need to validate that yourself. In relation to what's happening inside the pouch with the materials inside the pouch, the likes of extractables and leachables are very important to understand the integrity of the whole system, including the materials that are actually being used in both the product and the packaging. A lot of companies are starting to use or do the ENL testing, extractables and leachables testing, at a much earlier stage. And the reason for this is done is so that they have the final product materials and packaging materials at a much earlier stage, so they can make better decisions and earlier decisions to move the product and package along through its design verification. It's also done, say, like through the biocompatibility testing stage, and should also be done at post-marketing or surveillance stage. Again, it's a very important test to understand what has been extracted from your product, what has been given over to the customer, the patient at the end of the day. We have a huge amount of other tech talks available. All are available online. We have tech talks on accelerated and real-time aging, medical electrical equipment, distribution transportation testing, accelerated lifestyle testing or life cycle testing, lure connectors, design verification, sterile barrier integrity and string testing, again, which we have here today. And all of them are available either in video form or also in white paper form. Thank you for your time today.